Okay, so last Friday we started to talk about flowering plant reproduction, and today we're going to go into that in a lot more detail, especially as regards pollination, which is one of the most interesting topics in flowering plant reproduction. I hope you enjoy this part. And then we'll get into the life cycle, which is pretty similar to the one we talked about last time in pines and gymnosperms. So that'll be just a slight wrinkle on what you saw last time in the pines. There's a further reduction of the gametophytes, and there's this phenomenon of double fertilization you've heard about in lab that we'll get into a little bit more. And then finally we'll talk about the fruit, which is one of the most important innovations of flowering plants as regards seed dispersal. All right, so last time in talking about pollination, I mentioned that in flowering plants, unlike in gymnosperms where the pollen is transported directly to the ovule, in angiosperms the pollen is transported to the stigma or stigmatic surface of the pistil. And we have typically, most commonly, we have both pollen and ovules being produced in the same structure of the flower. And so there's a real potential for selfing to occur. And selfing just means pollen from the same plant fertilizing ovules of that same plant. So basically, the consequences are pretty extreme in terms of inbreeding and the potential for unmasking deleterious genes, alleles, by basically having homozygosity at those alleles or at those loci is really a problem. And that's the most immediate selective reason why inbreeding is deleterious to plants and other organisms. And especially in angiosperms, the potential for that is high. That is for selfing. So when we look back at the common ancestor of flowering plants, based on phylogenetic reconstructions and our understanding of the characteristics of modern lineages, it appears that this genetic mechanism, self-incompatibility here, was present in the common ancestor of all angiosperms that are alive today. And this system basically is a genetically based system that involves a single gene, the S gene, or S locus, as it's called. And there are a large number of allelic variants of this gene. There's strong selection for lots of diversity at this locus because basically any pollen grain, like the ones up at the top of that stigma that you see on the leftmost figure here, that have the same, or that share one of the alleles of that gene with the pistillate parent. What you can see here is a pistil, ovules down at the base, and you see the genotype at the bottom there, S1, S2 on the leftmost figure. And you can see that the pollen grains at the top have either S1 or S2. So if they share one of the S alleles with the sporophyte pistillate parent, then there'll be an antibody-antigen-like reaction that'll prevent that pollen grain from successfully growing a pollen tube down to the ovules. There are different ways that reaction can occur, different stages of development of the mature male gametophyte. But the main point is that this is a recognition of self. In contrast, this kind of antibody-antigen reaction we have in our own bodies against non-self, like an invading disease agent, this is a recognition of self. And if the pollen instead has an allele that differs from both of those in the pistil, as you can see on the three rightmost pollen grains up at the top, that have S3, S4, and S5 that aren't shared with the S1, S2 parent, then those pollen grains are not going to be subject to that sort of reaction, and they can grow down successfully to ovules. And so this kind of system is widespread in flowering plants, and it seems to be ancestral, but it's been lost a number of times. You might wonder why, since this would prevent not only selfing, but also inbreeding involving, say, your siblings, close relatives, and you can militate against that, depending on what alleles they end up with. But imagine a situation where pollinators are unreliable. If you're in an environment where you're relying on animal pollinators and they just don't happen to turn out that year, and imagine if you're an annual plant that just lives for 1 year and flowers and then dies. That's sort of the most extreme example where you have selection against this self-incompatibility, because it's better to inbreed than it is not to reproduce at all. And that's your only reproductive opportunity, then you better take it. So self-compatibility, as it's called, has evolved multiple times through selection for mutants that have knocked out this system. And but now I want to talk about mechanisms that can help to militate against selfing, but don't completely prevent selfing. And we often see these other mechanisms I'm going to talk about in self-compatible plants. And these self-compatible plants that can self but can outcross as well, they often have a bed hedging strategy, we could call it, where they'll outcross if it's possible, but if outcrossing isn't possible, then selfing will occur. All right, so one type of mechanism other than self-incompatibility that prevents selfing, self-fertilization, is either monoecious or dioecious. On the left we have a monoecious plant, on the right we have a dioecious plant. So these are just conditions where you have unisexual flowers on an individual plant, either staminate flowers that don't produce functional carpels, or pistillate plants that don't produce functional stamens. And in the case of a monoecious plant, you have both kinds of unisexual flowers on the same plant, usually in separate inflorescences. So it is still possible to have selfing. If a staminate flower ends up moving pollen to a pistillate flower on the same plant, that has the same genetic consequences as selfing within an individual flower. But that spatial separation and often the differential timing of maturation of the flowers of different expression on the same plant will tend to militate against selfing in those kinds of those plants. But selfing is possible again. But that spatial and oftentimes also timing difference in maturation of the flowers will tend to militate against selfing. But on the right we see a more extreme condition. Dioecious, where you only have one type of flower on individual plants. So you have one type of unisexual flower. So you either have staminate flowers like on the left in that willow there, or on the right we have pistillate flowers only in the same species of willow shown there. So in that case selfing is impossible, and that's as robust a system that prevents selfing as genetic self-incompatibility. But occasionally dioecious plants will produce a flower of the other expression, so rarely these plants can self. It can happen. But commonly this kind of system will evolve from a self-compatible ancestor when there's really strong selection for outcrossing. So there's no, and along that line, I should mention that there's no example that's yet been found where genetic self-incompatibility has been reconstituted once lost. It's a complex system, and it's one of these systems that once it's knocked out, it seems never to re-evolve. So there are other ways in which plants can enforce outcrossing, like dioecious there. All right, another type of mechanism to prevent self-fertilization we see in flowering plants that have bisexual flowers, ones that have function in the same, a staminate and pistillate function in the same flower, is spatial separation of the anthers and the stigma in the same flower. And that's not a hugely robust way to prevent selfing, but it's much better than, it militates against selfing to a much greater extent than if you had the anthers releasing pollen at the same level in the flower as the stigma, where you get immediate selfing. And on the left, this is an interesting system here that's present in a few groups. And usually there's only one floral morph. Either the anthers are held above the stigma or the stigma is held above the anthers in a given species. But in occasional groups, we've had the evolution of a system that Darwin studied where we have two different flower morphs that are present in different individuals. In one individual of the same species, you'll have a short-styled morph where the anthers are held high and the stigma is held low. And in the right example, you can see where we have a long-styled flower where the style is held high and the anthers are held low. And note how the position of the anthers on the rightmost flower is about the same as the height of the stigma on the one on the left. And similarly, the style on the right, the stigma there is at about the same level as the anthers on the left. And it turns out that there's a self-incompatibility system that only allows these two floral morphs to cross to one another. But at the same time, there's the placement of pollen on a pollinator, an animal that pollinates the flower, is going to be usually about the same with each visit to an individual flower. And so this will tend to enforce that compatible morphs will place pollen on compatible stigmas. But there's also this self-incompatibility system that enforces it as well. So it definitely will prevent pollen wastage at the same time that you have this kind of self-incompatibility system. But the main point I wanted to make here is just that holding the anthers at a different position in the flower than the stigma, that spatial separation, will tend to militate against selfing. Another way to militate against selfing in a bisexual flower that produces both functional stamens and functional carpels is not separation in space of the anther and the stigma, but separation in timing of the receptivity of the stigmas and the release of the pollen. And so this is a great case here in a magnolia flower, where on the left you can see the stigmas are exposed and receptive up near the top of the receptacle there. Before the anthers shed pollen, the anthers are down below. You can see they're tightly oppressed to the receptacle. Whereas on day 2, 24 hours later, the anthers have begun to open and shed pollen. So there's a period of about a day where outcrossing can occur without any competition from pollen from the same flower. Now, it could be that another flower on the same plant is releasing pollen at the same time that the stigmas are receptive at the beginning of that flower's period, but at least there's spatial separation there that would help to militate against selfing as well when it's happening in separate flowers. So this kind of system is really common, and oftentimes we get both spatial separation and timing separation in the same flower that will militate against selfing. And it's not always the case that the stigmas are receptive first and then the anthers are releasing pollen. Sometimes it's the other way around. And that tends to be something that's conserved within clades. So even within families of flowering plants, you sometimes get one or the other going first. Either the stigmas are receptive first or the anthers are releasing pollen first. So these are just some general mechanisms against selfing in case that are important when you have the potential for selfing being so enhanced in flowering plants, given their morphology of the flower. All right, so now I want to talk about the actual mechanism of moving the pollen between the stamen and the stigma. And in flowering plants, unlike in gymnosperms in general, wind is not nearly as important. I mean, there's roughly 20% of flowering plants, 1 in 5 species, is either wind or water pollinated by some physical force, unlike in gymnosperms where basically all but the cycads are wind pollinated. Cycads are insect pollinated. But in animal pollination in flowering plants accounts for about 80% of the species of flowering plants. And so this is going to involve a process known as coevolution, which I'm sure you heard about earlier in the course. And just to remind you, coevolution is a type of evolutionary interaction involving two organisms where an evolutionary response in one is followed by an evolutionary response to that response by the other. And this has been going on in flowering plants now for over 130 million years, where pollinators and flowering plants have been co-evolving. And it's led to some really remarkable and amazing changes in both partners. This is a great example here, one of the most famous in flowering plants that was first studied by Darwin back in the 1800s, involving the Madagascar star orchid shown here on the left. And orchids have an amazing array of pollinator specializations, but this one is sort of off the scale. You can see this pollen tube here that runs up to the back of the flower. It's around 8 inches long, and it's very narrow. The nectar in this tube accumulates down at the base, so any pollinator that's going to visit that flower and actually get a reward has to have a tongue long enough to go all the way down to the base of that nectar tube. And that seemed pretty unlikely, but Darwin made the prediction that someday someone would find a hawk moth that had a tongue that was just about perfectly the length of that tube, because if it was shorter, it wouldn't get the nectar reward, and if it was longer, its face wouldn't come into contact with the stamens or the stigmatic surface, and it couldn't be an effective pollinator. And in fact, about 2 decades, about 20 years after Darwin died, the animal on the right was found, which has been named Darwin's hawk moth because it was predicted by him, even though he never saw it. And it has a tongue that's perfectly fitting with that star orchid nectar tube, and it has been observed and even photographed unrolling this really long tongue and then poking it down into this nectar tube. And you can see, I think, that footage even online of one of these things visiting at night. All right, so the animal pollination, or biotic pollination as it's called, in flowering plants requires has two basic requirements. First I want to mention that biotic pollination is ancestral in flowering plants. Even though wind and water pollination do occur, they seem to be derived multiple times from animal pollination based on our understanding of the phylogeny of plants. And one of the most important, probably the most important component of biotic pollination is that the plant has to have some way to attract the pollinator. It has to have an advertisement or an attractant. And the most common type of advertisement is to have showy parts that stand out from the environment as being readily visible to the pollinator. And this can involve different parts of the flower, or even parts of the inflorescence that subtend the flower that aren't actually part of the flower. So it can involve showy petals, sometimes showy sepals. It can also involve showy stamens, and even bracts beneath the flower. In some plants, there's also the production of floral odors. And depending on the pollinator that the plant is co-evolved with, those will be odors that pollinator finds attractive, and can vary from anything from fruity and pleasant to our nose to really fetid and rotten. And talk about some of those examples. The plant on the right there, Amorphophallus titanum, is the Titan arum, is also called the corpse flower. Even though it's actually an inflorescence, it's the largest unbranched inflorescence in the world that's known. From Indonesia, it's grown in the Botanic Garden up here in Strawberry Canyon, and occasionally you'll see announcements on the Berkeley homepage when it opens, and it's absolutely spectacular. If it does happen while you're here at Cal, you should definitely go up and see it on the day that it opens, because that's the day that it produced the most spectacular odor, which is that of a rotting corpse. So it's pretty extreme. It attracts beetles and flies that regularly visit cadavers. So it's a pretty amazing plant. Another component of biotic pollination that's usually present, understandably, is some kind of a reward. If you just attract the pollinator with the promise of a reward and there's nothing there, a smart pollinator is going to learn its lesson and you're not going to get regular visitation. So a reward for the pollinator is often presented, and as you can imagine, the ancestral reward in flowering plants, the most primitive reward, was likely pollen, because pollen had to be produced anyways for reproduction to occur. And it actually is very nutritious. Remember, it's the male gametophyte, so it's a little organism that's immature at that stage, but it has a lot of protein content and fat. It's actually a rich source of nutrients. But think about the plant side of that equation. It's very expensive to the plant. And we've seen numerous times the evolution of nectar as a cheaper reward. We might think of nectar as more desirable, it's sweet, tastes good, but it's just sugar water. It's pretty devoid of nutrition. So it's a cheap way of satisfying a pollinator. I mean, it can be nutritious. There are sometimes amino acids in nectar. It can be nutritious to the pollinator and critical to the pollinator's survival, of course, but it's relatively inexpensive for the plant compared to pollen. And it doesn't interfere with, you're not actually losing some of your reproductive output. Whereas if your pollen grains are getting eaten, it's basically a trade-off, moving some of them and losing the rest to your pollinator. So if nectar is a reward, then pollen is going to be carried incidentally, and or it can be carried. Some pollinators will collect both pollen and nectar, female bees especially. But animal pollinated plants have sticky pollen, so the pollen needs to be able to adhere to the pollinator, and it's typically clumped, oily, or sticky, and adheres readily to fur, feathers, anything, and can be moved around that way. And it's often sculptured, as I mentioned earlier. Rather than having a smooth wall, it's sculptured, and the oil and all can be held in among these spindules and such. So that's very typical of an animal pollinated type of pollen. Also, remember that if, in general, both in terms of presenting the advertisement and the reward, the flower opening, the timing of the flower needs to be synchronized with the activity time of the pollinator. And we'll see some examples of that in a moment. But obviously, a plant that's not in synchrony with the activity time of its pollinator is not going to be pollinated effectively. All right, so by far, the most important pollinators worldwide, in terms of we look at all the diversity of flowering plants, are bees. Bees don't visit all plants, but I'm saying that the majority of plant species, bees are important pollinators. And bees are great pollinators because they're intelligent, as insects go. They learn well, and they can learn to work flowers very well. They tend to, well, we talked about earlier that their visual range is close to our visual range. It's offset slightly towards shorter wavelengths. So reds and oranges look like black to them and are not attractive. But a lot of blue, basically yellows to blues, are very nicely visible to bees, as is the UV end of the spectrum. And I mentioned that earlier on, that they can see patterns that we can't make out in the UV end of the spectrum. Bees also have really good pattern recognition, and oftentimes bee-pollinated plants will have things like bullseyes, like you see up there on the top, or they'll have guidelines that act like the strip or the lines drawn on a landing strip at an airport that help to orient them towards the business part of the flower at the center. And so they have really good pattern recognition. They remember things, and they collect both nectar. Well, nectar is the main thing they're after if they're a male bee, but female bees are collecting pollen and nectar typically. All right, so bees are also, even though they're smart, they're sometimes deceived, and it's possible to deceive a bee. There's deception is one of the common features of orchid pollination. I showed you an orchid that actually got a reward, that star orchid, but many orchids, thousands of species of orchids, do not provide a reward for their pollinator, and so they have what we call deceit pollination, where there's a promise of a reward that's never provided. And some of this deceit is so good that it can even fool bees, and they'll repeatedly visit the same flower, not realizing they're being deceived constantly. The flip side of this is that there are also insects that are nectar thieves, and so sometimes plants are actually robbed of nectar or pollen without being pollinated. So there are cheaters in both directions in pollination. But in general, we do see a reward that's given, and pollinators do provide good service. But it's important to realize that at least a lot of orchids are cheaters with regard to their pollinators, and they have very specialized pollination. Orchids do in general. This is a great case in point. These bee orchids produce flowers, maybe not to us, but to some extent, but to a male bee, these look like female bees. And more than that, they actually produce pheromones that mimic the sex pheromones of female bees. And this is so successful at tricking male bees into trying to copulate with the flowers that it's had a huge, these are hugely diverse. These bee orchids, as you can see, come in a variety of forms and colors, and they've diversified extensively because of this very successful deceit. So I'll show you one more time. This flower down here on the lower right is a bee orchid, which I think you can see looks a lot like a bee there. Here's another one. And the male bee is going to come in here. And the stamen and the stigmatic surface are right under, right above its head. So it's copulating with the flower. It's attempting to, but it doesn't quite work out. And so it's sort of frantically trying to copulate, but it can't do it. And in the process, you can see there the pollinium mass is now on its head, and now it's flying away. And it'll take that to the next flower, do the same thing, and deposit that whole pollinial mass on the stigmatic surface of the next flower. So really,